In emergency medicine, you stop measuring age in years relatively early in your career. You start measuring it in systems, in the state of the vasculature, the resilience of the myocardium, the speed of metabolic recovery, the degree to which the body's compensatory mechanisms remain intact and responsive under physiological stress. And what that systems-based perspective reveals, with a consistency that never quite loses its power to surprise, is that chronological age is one of the least reliable predictors of biological state available to you. Two patients, both 67, one whose biology is performing at 50, one whose biology has already reached 80. The calendar does not explain the difference. The science increasingly does. The question of why some people age slower than others is no longer a question that belongs exclusively to philosophy, genetics, or speculation. It is a question that the molecular biology of the past 2 decades has begun to answer with a precision and a mechanistic clarity that has transformed longevity from a subject of popular curiosity into a rigorous scientific discipline. The answers that have emerged are not simple, and they do not reduce to a single gene, a single lifestyle variable, or a single molecular pathway. But they converge on a set of biological mechanisms, processes operating at the cellular and subcellular level that explain with increasing completeness how the body ages and why that process unfolds at dramatically different rates in different people. More importantly, they identify which of those mechanisms are fixed and which are modifiable. A distinction with profound practical implications for everyone on the far side of 40. Understanding why some people age slower requires understanding what aging actually is at the biological level, and that understanding begins not with years, but with cells. The scientific framework that has most productively organized our understanding of the biology of aging is what researchers call the hallmarks of aging, a set of cellular and molecular processes, first systematically described in a landmark paper published in Cell in 2013 by Lopez-Otin and colleagues, and subsequently updated in 2023, that represent the primary mechanisms through which biological aging occurs across species. The hallmarks are not a list of symptoms, they are a description of the underlying processes that cause the functional decline associated with aging. And they operate simultaneously, interacting with and amplifying one another in a network of biological deterioration that the researchers characterize as both the cause and the consequence of the aging phenotype. The original framework identified 9 hallmarks. The 2023 update expanded this to 12, incorporating mechanisms whose significance had been established by the subsequent decade of research. They include genomic instability, the progressive accumulation of DNA damage that the cell's repair mechanisms become less able to fully correct over time. Telomere attrition, the shortening of the protective caps on chromosomes that occurs with each cell division, and that below a critical threshold triggers cellular senescence or apoptosis. Epigenetic alterations, changes to the pattern of gene expression that occur with age independently of changes to the DNA sequence itself, and that progressively dysregulate the coordinated gene expression on which healthy cellular function depends. Loss of proteostasis, the degradation of the quality control systems that maintain the proper folding, function, and clearance of proteins, leading to the accumulation of misfolded and dysfunctional proteins that impair cellular operation. Disabled macroautophagy, the decline of the cellular recycling system that degrades and repurposes damaged organelles and protein aggregates. Deregulated nutrient sensing, alterations in the signaling pathways that communicate the body's nutritional and energy status to cells, pathways whose dysregulation drives many of the metabolic features of aging. Mitochondrial dysfunction, the progressive decline in the efficiency and integrity of the cellular energy-generating organelles. Cellular senescence, the accumulation of cells that have permanently exited the cell cycle and that secrete a cocktail of inflammatory molecules called the senescence-associated secretory phenotype that damages surrounding tissue. Stem cell exhaustion, the depletion of the regenerative capacity that tissue-specific stem cells provide. Altered intercellular communication, the progressive dysregulation of the signaling networks through which cells coordinate function across tissues and organs. Chronic inflammation, the low-grade persistent inflammatory state that characterizes biological aging and that contributes to virtually every age-related disease. And dysbiosis, the age-related alteration of the gut microbiome composition that has downstream consequences for immunity, metabolism, and systemic inflammation. The significance of this framework for understanding why some people age slower is not merely descriptive, it is mechanistic. People who age more slowly are, at the biological level, people in whom these hallmark processes are progressing more slowly, in whom DNA damage is accumulating less rapidly, telomere attrition is less pronounced, epigenetic drift is better controlled, senescent cell accumulation is lower, mitochondrial function is better preserved. The science has moved far enough to begin identifying with some specificity which biological and behavioral variables determine the rate of each of these processes. One of the most consequential methodological advances in longevity science over the past decade has been the development of tools capable of measuring biological age directly, tools that provide us a readout of how far the hallmark processes have progressed in a given individual, independent of their chronological age. The most powerful of these tools is the epigenetic clock, and its development has transformed the scientific study of aging in ways whose implications are still unfolding. Epigenetic clocks are based on the measurement of DNA methylation patterns, chemical modifications to DNA that regulate gene expression without altering the underlying genetic sequence. These methylation patterns change with age in a highly predictable manner across the genome, at specific sites that have been identified through machine learning analysis of large datasets, comparing methylation patterns with chronological age and health outcomes. The first generation of epigenetic clocks, developed by Steve Horvath at UCLA and published in Genome Biology in 2013, could estimate chronological age from methylation data with a median error of approximately 3.6 years across multiple tissue types, a precision that established DNA methylation as a more reliable molecular record of biological time than any previously available marker. Subsequent generations of clocks, most notably the PhenoAge clock developed by Morgan Levine and colleagues, and the Grimmage clock, were trained not simply to predict chronological age, but to predict mortality and disease risk, to measure not how old the cells appear, but how old they are behaving in terms of their functional and clinical consequences. These second-generation clocks have established that biological age, as measured by epigenetic methylation, diverges from chronological age in consistent ways that are associated with health outcomes. People whose epigenetic age is younger than their chronological age, described as epigenetic age deceleration, have lower rates of cardiovascular disease, cancer, and neurodegenerative disease, and longer survival, compared to people of the same chronological age whose biological age is older or equivalent. The critical finding for the practical question of why some people age slower is that epigenetic age is not fixed by genetics. Research examining identical twins who share their DNA sequence entirely has demonstrated that epigenetic age can diverge substantially between twins over decades of adult life, with the divergence explained predominantly by differences in lifestyle, environmental exposures, stress, and health behaviors rather than by genetic factors. A study published in Aging examining epigenetic age in identical twin pairs found that the twin who exercised more, smoked less, maintained healthier body weight, and experienced lower chronic stress had measurably younger epigenetic age than their genetically identical counterpart, sometimes by a margin of several biological years. Genetics provides the starting position, biology provides the trajectory, and the trajectory, it turns out, is substantially under the influence of factors that are modifiable. Telomeres, the repetitive DNA sequences that cap the ends of chromosomes and protect them from degradation and end-to-end fusion, represent one of the most extensively studied mechanisms of aging. And their biology illustrates with particular clarity the interplay between fixed genetic factors and modifiable environmental and behavioral influences that characterizes the longevity science field broadly. With each cell division, telomeres shorten because the DNA replication machinery cannot fully replicate the terminal ends of linear chromosomes. Below a critical telomere length threshold, the cell enters a state of permanent growth arrest, senescence, or undergoes programmed cell death, apoptosis. This progressive telomere shortening is one of the primary molecular mechanisms limiting cellular replicative capacity and driving the accumulation of senescent cells that characterizes aging tissue. Telomere length in leukocytes, white blood cells accessible from a standard blood sample, is associated in large epidemiological studies with biological age, disease risk, and mortality. And individuals with longer telomeres at a given chronological age have, as a group, better health outcomes and greater longevity. The rate of telomere attrition, however, is not fixed. It is influenced substantially by oxidative stress, the accumulation of reactive oxygen species that damage DNA, including telomeric DNA, and that accelerates telomere shortening beyond the baseline rate imposed by replication. Chronic inflammation similarly accelerates telomere attrition through inflammatory cytokines that increase cell turnover and oxidative burden. Chronic psychological stress operating through cortisol and the HPA axis promotes both oxidative stress and inflammation and is associated in multiple studies with accelerated telomere shortening, with one of the most striking demonstrations being research by Alyssa Eppel and colleagues published in the Proceedings of the National Academy of Sciences, showing that mothers of chronically ill children, a well-characterized high-stress population, had telomeres equivalent to those of women a decade older than their chronological age, with the degree of shortening correlating with the duration and subjective severity of the caregiving stress. The converse is equally well documented. Regular physical activity is associated with longer telomeres and reduced telomere attrition rates across multiple large studies, with the Leicester Biobank research demonstrating that highly active individuals had telomeres corresponding to a biological age approximately 10 years younger than sedentary individuals of the same chronological age. Adequate sleep, dietary antioxidant intake, and practices that reduce chronic psychological stress, including mindfulness-based interventions, whose effects on telomere biology have been examined in randomized controlled trials, are each associated with attenuated telomere shortening. The rate at which this fundamental molecular clock of aging progresses is, to a degree, the early telomere research did not anticipate, a behavioral variable. Of the 12 hallmarks of aging, mitochondrial dysfunction has attracted increasing attention in longevity science as both a primary driver of biological aging and a target whose modifiability, through specific interventions, is well supported by both mechanistic and clinical research. Mitochondria, the organelles responsible for generating the majority of the cell's energy through oxidative phosphorylation, decline in both number and function with age. The efficiency of the electron transport chain decreases. Mitochondrial DNA accumulates mutations at a higher rate than nuclear DNA, lacking the equivalent repair mechanisms. Damaged mitochondria accumulate within cells as the mitophagy pathway, the quality control process that identifies and degrades dysfunctional mitochondria, becomes less effective. The net result is a decline in cellular energy production capacity that affects every energy-demanding tissue in the body, with the highest energy demand tissues, the brain, the heart, the skeletal muscle, most severely affected. Mitochondrial dysfunction does not simply reduce energy availability. It increases the production of reactive oxygen species which damage proteins, lipids, and DNA throughout the cell, accelerating multiple other hallmark processes simultaneously. The relationship between mitochondrial dysfunction and the other hallmarks is therefore not merely one of parallel progression. Mitochondrial decline acts as an amplifier of the broader aging process, with its deterioration accelerating genomic instability, epigenetic dysregulation, and cellular senescence through the oxidative damage it generates. The relevance of this to why some people age slower is the well-established relationship between regular physical activity and mitochondrial biogenesis, the formation of new functional mitochondria in response to the energetic demands placed on muscle cells during exercise. Exercise activates PGC-1-alpha, a master regulator of mitochondrial biogenesis, which drives the production of new mitochondria, the upregulation of antioxidant defense systems, and the improvement of mitochondrial quality control mechanisms. The effect is not confined to skeletal muscle. The systemic signaling environment created by exercising muscle, including the myokine and hormonal signals described in the context of the longevity signal discussion, influences mitochondrial function in distant tissues, including the brain and the cardiovascular system. Research examining mitochondrial density and function in highly active older adults compared to sedentary age-matched controls has found differences in mitochondrial capacity equivalent to those observed between individuals 20 to 30 years apart in chronological age. One of the most compelling direct demonstrations that the molecular biology of aging in specific cellular compartments is genuinely modifiable through the right physiological stimuli. One of the most important conceptual advances in longevity science over the past decade has been the characterization of what researchers call inflammaging, the chronic, low-grade, sterile inflammatory state that accumulates with biological age and that functions as both a consequence and a driver of the hallmark aging processes. Understanding inflammaging is central to understanding why some people age faster than others, because the degree to which it develops and progresses is one of the most variable and most modifiable dimensions of biological aging. Senescent cells are a primary driver of inflammaging. As cells throughout the body reach their replicative limit or are damaged beyond the threshold that triggers senescence, they exit the cell cycle permanently but do not die. Instead, they remain metabolically active and begin secreting the senescence-associated secretory phenotype, a complex mixture of inflammatory cytokines, matrix metalloproteinases, growth factors, and other signaling molecules that have paracrine effects on neighboring cells, promoting inflammation in surrounding tissue, degrading the extracellular matrix, and inducing senescence in nearby cells through a process called bystander senescence. Small numbers of senescent cells are managed effectively by the immune system, which under healthy conditions identifies and clears them. As immune function declines with age, a process called immunosenescence, the clearance becomes less effective, senescent cell burden accumulates, and the inflammatory output of the senescent cell population increases progressively. The result is a systemic inflammatory background that, while far below the levels associated with acute infection or acute inflammatory disease, is chronically present, chronically damaging to endothelial and parenchymal tissue, and chronically accelerating of the other hallmark processes. Elevated circulating levels of inflammatory markers, IL-6, TNF-alpha, C-reactive protein, in the absence of acute illness are associated in large epidemiological studies with accelerated biological aging, higher risk of cardiovascular disease, cancer, dementia, and all-cause mortality. They are also critically associated with the same lifestyle variables that predict telomere attrition and epigenetic age acceleration, sedentary behavior, poor sleep, chronic psychological stress, and diets high in ultra-processed foods. The people who age slower are, at the level of this mechanism, people in whom the senescent cell burden is lower, the immune clearance of senescent cells is more robust, and the chronic inflammatory background is better controlled. Not because they lack the biological processes that generate senescent cells, but because the rate of senescent cell accumulation is slower, and the clearance mechanisms remain more functional for longer. Among the most intellectually productive areas of longevity biology over the past 3 decades has been the study of nutrient-sensing pathways, the molecular systems through which cells detect and respond to the availability of energy and nutrients, and whose modulation has been shown in organisms from yeast to mammals to have profound effect on lifespan and healthspan. The 3 most studied of these pathways are the mTOR pathway, the insulin and IGF-1 signaling pathway, and the AMPK pathway, and their interrelationship constitutes one of the central regulator axes of cellular aging. mTOR, the mechanistic target of rapamycin, is a kinase that integrates signals from nutrient availability, growth factors, and cellular energy status to regulate protein synthesis, cell growth, and autophagy. When nutrient and energy signals are high, mTOR is activated and promotes anabolic processes, growth, protein production, cell proliferation. It simultaneously suppresses autophagy, the cellular recycling process that degrades damaged proteins and organelles. Chronically elevated mTOR activity, characteristic of the nutrient surplus environment of modern life, suppresses the cellular maintenance and quality control processes that protect against the accumulation of cellular damage, accelerating multiple hallmark aging processes in parallel. AMPK, in contrast, is activated when cellular energy is low during fasting, caloric restriction, or vigorous physical activity, and promotes precisely the cellular maintenance processes that mTOR suppresses: autophagy, mitochondrial biogenesis, and the repair of cellular damage. The inverse relationship between mTOR and AMPK activity is one of the primary molecular mechanisms through which caloric restriction extends lifespan in model organisms, and through which the metabolic effects of physical activity produce benefits that reach far beyond the immediate energetic expenditure of the exercise itself. The longevity implications of this pathway biology are specific and actionable. Chronic caloric surplus and continuous eating patterns characteristic of the modern nutritional environment maintain chronically elevated mTOR activity and suppressed AMPK, creating a molecular environment that accelerates cellular aging. Periods of fasting or caloric restriction, independent of total caloric intake, activate AMPK and suppress mTOR, promoting the cellular maintenance programs that slow the accumulation of biological damage. The research on time-restricted eating in humans, while still maturing, has demonstrated effects on autophagy markers, inflammatory markers, and cellular aging biomarkers that are consistent with the pathway biology established in model organisms, suggesting that the meal timing patterns described in the metabolic health context have longevity implications that extend beyond glucose regulation into the fundamental biology of cellular aging itself. The longevity science, taken in its entirety, does not point to a single intervention, a single gene, or a single molecular target as the explanation for why some people age slower. It points to a set of overlapping, mutually reinforcing biological processes that are collectively influenced by a consistent set of variables, and the convergence of evidence across these processes on the same variables is one of the most striking features of the field. Regular physical activity, and specifically the combination of resistance training for muscle preservation and cardiovascular exercise for mitochondrial and cardiovascular health, attenuates telomere attrition, decelerates epigenetic aging, promotes mitochondrial biogenesis, reduces senescent cell burden through immune and anti-inflammatory mechanisms, activates AMPK and the cellular maintenance programs associated with longevity, and reduces the chronic inflammatory background of aging across multiple convergent pathways. The magnitude of biological age difference associated with lifelong physical activity, measurable across telomere length, epigenetic clocks, mitochondrial density, grip strength, and inflammatory biomarkers, represents one of the largest effects on biological aging that the research has identified from any single modifiable variable. Sleep at sufficient duration and quality is the period during which the majority of cellular repair processes associated with the hallmarks operate. Autophagy peaks during sleep, DNA repair is most active during sleep, inflammatory resolution occurs during sleep, and the glymphatic clearance of neurotoxic protein aggregates from the brain occurs almost exclusively during deep sleep. Chronic sleep insufficiency accelerates epigenetic aging, elevates inflammatory markers, shortens telomeres, and impairs the immune clearance of senescent cells, with an effect size in the longevity biology literature that places it among the most consequential modifiable determinants of biological age. Chronic psychological stress, operating through sustained cortisol elevation and HPA axis dysregulation, accelerates telomere attrition, promotes epigenetic age acceleration, drives inflammaging, suppresses immune function, and impairs the cellular maintenance processes associated with longevity, with effect sizes that are, in the research literature, among the largest associated with any psychosocial variable on biological outcomes. And diets, specifically diets characterized by high nutrient density, high fiber content, minimal ultra-processed food, and meal timing patterns that allow periods of low insulin and mTOR activity, influences biological aging through every one of the pathways described above, from telomere protection through antioxidant intake to AMPK activation, through fasting periods to inflammaging reduction through gut microbiome support. The people who age slower are not, in most cases, people who have found a single secret. They are people in whom multiple biological aging processes are progressing more slowly, because multiple modifiable variables that influence those processes are being managed well, often without explicit knowledge of the molecular biology involved, but with a consistent pattern of behavior that the molecular biology, when examined, explains with remarkable completeness. The science of why some people age slower is no longer a mystery. It is a set of mechanisms increasingly well characterized that describe precisely how biological time accumulates in human cells, and precisely which decisions, repeated daily over decades, determine how quickly that accumulation proceeds. That is not a peripheral footnote to the clinical picture. In the emergency department and beyond, it is increasingly central to it.